of all, I would like to thank the organizing committee for giving me the opportunity to present this work. And this presentation is based on a recent publication, and it's about the promising effects of melatonin on radiated human breast cancer cells. Breast cancer is a classic model of hormone-dependent malignancy, and for this reason, one of the main objectives of the endocrine therapy has always been to neutralize the effects of estrogens on the breast. On the one hand, we have drugs that interfere with the effects of estrogens, which are called the selective estrogen receptor modulators. And on the other hand, drugs that interfere with the synthesis of estrogens, the selective estrogen enzyme modulators. Melatonin is a natural hormone which is mainly produced in the pineal gland during the night and has both anti-estrogen and also anti-aromatase properties. The sequencing of chemotherapy, radiation, and also endocrine therapy is always a challenge for the oncologist. And also an important question is whether to combine endocrine therapy and post-surgery radiotherapy in order to select the best treatment approach. Since melatonin exerts oncostatic actions both in tumor cells and also in mammary tumors, the objective of this work was to investigate the effects of this hormone on radiated human breast cancer cells. So with this aim, we treated MCF7 cells with different melatonin concentrations for 7 days, and then cells were irradiated at different doses in a range between 4 and 12 grays. After that, after different incubation times, depending on the experiment, cells were used to study cell proliferation, cell cycle phase distribution, and also the gene expression of two of the main proteins involved in DNA repair mechanism. When we study the effects, when we study the effects of melatonin on MCF7 cells, we found that incubation with different concentrations of this hormone led to a significantly decrease in cell proliferation compared to control cells, both at 3, 4, or 6 days of incubation. And the greatest inhibition was found with melatonin at physiological concentration, which is 1 nanomolar. Then we studied the effects of ionizing radiation on MCF7 cells, and as expected, cell proliferation decreased in a dose-dependent manner compared to control non-radiated cells. For example, for an 8 grays radiation dose, the decrease in the survival fraction was about 40% at 3 days and almost 80% at 6 days post-radiation. And this is the dose we use for the rest of the work. Next, we study the effects of melatonin pretreatment on proliferation of radiated human breast cancer cells. And what we found was that incubation with different concentrations of melatonin led to a significantly decrease in cell proliferation compared to control radiated cells. In this study, we also tried to investigate if there was any difference when melatonin was present in the culture media after the irradiation, which are the striped columns on the right. But as we can see on the graph, there were no significant differences. So these results suggest that melatonin may enhance the inhibitory effects of ionizing radiation on MCF7 cells. It has been described that the antiproliferative effects of melatonin are due in part to its ability to modulate cell cycle. So when we study cell cycle phase distribution in MCF7 cells, we found that cells that were pretreated with melatonin at physiological concentrations, there was a significant cell cycle arrest in the G1 phase, thus decreasing the percentage of cells in the S and also in the G2M phase. With radiation alone, there was a significant cell cycle arrest in the G2M phase compared to control non-radiated cells, and the percentage of cells in the G1 and also in the S phase was lower than that of the non-radiated cells. And when cells were pretreated with melatonin and then irradiated, we found that the percentage of cells present in the G2M phase cell cycle arrest was lower, and there was an increase in the number of cells present in the G1 phase. Also, the number of cells in the S phase was lower too, So this may be the reason to explain the observed effects of melatonin that was shown in the previous slide. It is known that ionizing radiation induces high levels of DNA damage, especially double-strand breaks, which accumulation is very toxic, dangerous for the cell, and needs to be repaired. Depending on the extent of damage, cells may respond in two different ways, and both through activation or upregulation of p53. In general, cell cycle checkpoints are activated to allow DNA damage repair. So genomic integrity is restored and cells can survive. But when the damage is very extent, or where the repair is not possible, cells will be eliminated by cell death programs. In mammalian cells, there are two main pathways to repair double-strand breaks, and the choice of one of them depends on the phase of the cell cycle. For example, in the case of homologous recombination, which is maybe the most important one because it's an error-free mechanism, only occurs in the S and the G2S phase. And in this pathway, one of the most representative proteins is the RAD51, which is the most downstream one. So what we did was to study the effects of radiation and melatonin pretreatment on RAD51 gene expression by RT-PCR, and what we found was that radiation alone decreased the gene expression of RAD51 compared to control non-radiated cells, but when cells were pretreated with melatonin at different concentrations, there was a significantly decrease in RAD51 gene expression compared to control radiated cells. So if homologous recombination is partially inhibited by melatonin pretreatment, that may enhance the response of the tumor cell to ionizing radiation. The other pathway involved in double-strand breaks repair is the non-homologous edge joining, which can be activated during the whole cell cycle. And also we choose one of the most representative proteins in this pathway, which is the DNA PKC. And when we study the effects of radiation and melatonin on DNA PKC gene expression, we found also that radiation decreased its gene expression compared to control radiated cells, and once again, melatonin pretreatment at different concentrations led to a significantly decrease in DNA PKC gene expression. So based on these results, we postulate that the effects of melatonin are due in part to the impairment of double-strand breaks repair mechanism. Finally, we study the effects of ionizing radiation and melatonin on p53 gene expression. As we all already know, p53 is a tomosuppressor that activates several genes involved in different pathways such as cell cycle repair, cell cycle arrest, DNA repair, but also apoptosis. As expected, radiation increased p53 gene expression compared to control non-radiated cells, and once again, cells that were pretreated with melatonin was p53 gene expression was significantly upregulated. So based on our findings, we suggest that melatonin pretreatment sensitizes breast cancer cells to the effects of ionizing radiation. First, by decreasing MCF7 cell proliferation, especially at physiological concentrations, and also by inducing cell cycle arrest at the G1 phase, thus decreasing the percentage of cells that are present in the S phase, which is the phase where the cells are more radioresistant. And finally, by downregulating the expression of RAD51 and DNA PKC, which are two proteins involved in double-strand breaks repair mechanism. And all these actions seems to be dependent through an activation of p53. And as a final conclusion, these results open a new research line where melatonin maybe could be used in the future as a therapeutic agent, maybe as a part of an adjuvant therapy, to increase, in order to increase the sensitivity of tumor cell to ionizing radiation, or even to chemotherapy. And finally, I'd like to thank all the people that is part of my research group, and thank you very much for your attention.